What I should have is a sign like over my for my person that says like, please wait. This meeting is about to start or something. Anyway, let's go ahead and get started. The popcorn up. See, there you go. As soon as I say it, a new person joins. So hi everybody. I'm Peter Schwartz. I'm the director of the Bioethics and Subject Advocacy Program of the Indiana CTSI. And I'm also lucky to be the director of the IU Center for Bioethics. It's great to have everybody here. Our speaker today is TJ Kasperbauer, who is a postdoctoral fellow at the IU Center for Bioethics and a member of the Bioethics Subject Advocacy Program as well. We are thrilled to have this talk. We've actually, it brings together a couple things that we've been looking at in these TREATS talks about data and ethics around data, and also around what we're doing in COVID and some of the challenges of COVID and the very, very clear importance of public health in this time period. So I'm thrilled that TJ has agreed to talk about this topic. The idea of TREATS talks are to be very useful to non-specialists in ethics. They're not meant to be sort of ethics cutting edge. They're meant to be more introduction to key issues surrounding an important topic, a topic that might be arising in actual research for people. And we do hope that for you, our attendees, some of the topics we talk about, that TJ will talk about today about data and public health and secondary use of data will apply to your research, even if it has nothing to do with public health or the cases he talks about. So we're thrilled to have you, thrilled to have TJ here. And as I said, all these talks are actually recorded and are available online soon after the talk, along with materials that the speaker may mention. At the IU Center for Bioethics website, which is now at the wonderful bioethics.iu.edu website. And I'll actually put in the chat, I'll put a link to the TREATS talk section of our website, which is also the section for the BSAP group. Anyway, with no further ado, I will give you Dr. Kasperbauer. Thanks, TJ.
Thank you, Peter. Let me check first. Can you hear me all right? Okay. Let me share my screen. Which screen are you seeing?
It is perfect. We are seeing the screen we should see. We are seeing the audience screen. Thanks.
Gotcha. Okay. So thank you all for being here. We are, of course, coming up on 1 year of COVID in Indiana specifically, but COVID has been around for over a year, of course. And so now we have lots of COVID data, and there's been a lot of discussions in the research community, as well as internally in BSAP and at IU, of how to use this data responsibly, what sorts of restrictions might there be or should there be, and what's the best way of optimizing the use of this data. There's also lots of concerns we've been having internally about some privacy issues potentially. So lots of COVID programs, COVID testing programs have been mandatory for employment, mandatory for, say, coming on a university campus. And so that raises lots of questions about what we should do with these data, these specimens going forward. A lot of the attention so far, at least among the research community or research ethics community, has been how do we use research data for public health purposes. My talk today, since we're sort of looking at potentially the second or third phase of COVID, sort of going the other direction. So we have all this data that was collected for public health purposes, and now what do we do as researchers or research-wise going forward? So in traditional TREATS format, I'm going to talk about some of the relevant regulations, and then I'm going to conclude with some comments on the potential ethical issues that may arise from using existing COVID data and specimens. I'm going to reverse sort of the format, typical format. I'm going to talk about first some of the documents I looked at. There are a lot of very useful guidance frameworks that have been put out there. I'm going to be mainly focusing on that last one on the slide in front of you, the Common Rule guidance. So for those of you who don't know, the Common Rule is the main federal regulation governing research in the US. That's where you're going to find a lot of the most relevant and helpful guidance for research and research ethics in the United States. Also, though, there have been a lot of interesting commentary. There's been a lot of interesting commentary on the research ethical issues with doing research during a pandemic and specifically for COVID. A lot of it is just adapting sort of traditional, well-known public health frameworks to COVID. And I know Peter said this is not supposed to be cutting edge, but now that we are a year into this, it's almost not even cutting edge anymore. There's been a lot of sustained discussion now, sustained attention on ethical issues, and there's a lot of interesting stuff to look at. I'm not going to be using any of these sources or citing them directly or specifically, but it did inform my analysis over the next 20 minutes. So the sort of start and the end to this topic is the question, can we use existing biospecimens for research? And if anyone was at Peter's last talk or at my talk, I think 2 years ago, you would know that there was a lot of discussion around this in the revisions to the Common Rule a couple of years ago, that we have all these de-identified biospecimens hanging around. Shouldn't we do more to protect them? And the short answer is, according to current regulations, de-identified biospecimens fall outside of research regulations, which mean they can be used without consent and without review. But that's really just a framework or really just a foundation, sort of the first step in talking about whether you can use COVID data for secondary purposes. There's a lot more that can be said. So one thing that I'm not going to go into too much detail on because I'm not an expert in this area is even though you can use de-identified, say, COVID specimens like a saliva test or a blood test for research, it's another question whether public health authorities can actually give you that data. I'm not as much of an expert on what the restrictions are for public health authorities, so I'm not going to be commenting on that as much. But in general, it's still the case that if you have access to de-identified biospecimens, say after they've been used in a diagnostic test for COVID, you can use it for secondary research without many other restrictions on that. Another relevant question, though, is what consent forms say. So there's a lot of variation in the types of COVID testing people have received. You may have been in a clinic, you may have been, say, in a parking lot. It may have been as part of a study. And the consent practices and consent agreements are going to change, or it has implications for how you can use that data for those specimens for secondary purposes. So when thinking through the possibilities, I guess, for using COVID-19 data for secondary research purposes, I tried to identify what are the most common ways researchers may be using data or maybe thinking about using COVID data that they do have access to. And so I wanted to outline some of these right away. And in case people don't see their own plans in this, maybe it would be good to bring that up in the Q&A. But I wanted to outline this because there's going to be 3 routes I identify, and they all have different implications for secondary use. So the first one is more along the lines of that picture I just showed you. And I think this is the most common. I was trying to find data on this, but I could not. You go to get a COVID test, you go to get a PCR test, the nasal swab, and it's just for a public health purpose. It's just for tracking an infection, controlling the infection, and there's no research as part of that, and that's very upfront. So it's just for a public health purpose. That's why you're giving the data, and nobody else involved is thinking about research from the very start. Second one, which people here may be more familiar with, is where there is combined public health and research purposes from the start, where, say, a public health authority at the state or local level has partnered with an academic medical center or some other research institution. So here, for example, my understanding is this is how it's been done with the Fairbanks School of Public Health and IU, working with county and state public health officials, where from the very start, they're collecting specimens, both for the public health and sort of diagnostic purposes and for additional research. And that has implications for secondary use because you're already telling people that they're consenting to research. And we're going to talk about that more in a bit. The third one I think is really interesting. It's the one that's received the most attention. So I'm not going to say too much about it, but I would say this is where most ethicists or people in the research community have been concerned over the last year. This is where we have some sort of research data already existing, and now we see some potential public health purpose. And so we alter the data, or we use it for a secondary purpose, which is public health. And the question is, now a year later, we may want to use that data again for research purposes. And it's unclear what we can do with that. So the example a lot of people have been focusing on is the Seattle flu study, where researchers recruited participants to a study on infectious diseases in the Seattle area 2 falls ago. And then when COVID hit, they said, hey, couldn't we use some of this blood we've collected to see whether we have COVID here? And initially people said no, because people had consented to research. The authorities, the regulators said no, but then they realized, no, this is an urgent public health crisis. We have the authority to use these specimens and look at them for public health reasons. We don't need to go back to the participants and get their consent. And so they did that, and that's how people first discovered that there had been COVID spreading around the Northwestern region 2 falls ago. And that brought up a lot of debates, or a lot of disputes anyway, about how you might be able to revise consent forms, or how you might be able to revise data to use it for secondary or tertiary purposes. Because in this case, the sort of identifying information would have changed, and you're going to have to go back and look and see what exactly did people agree to in the consent form. Is this directly contradictory, and does that even matter, given that's for public health purposes?
That's a point of information. You just said that COVID was circulating in Seattle 2 falls ago. You don't mean that, right?
It was. So I don't know the exact month, but I want to say like December. So they were able to retrospectively. So this was last April, where they were having this conversation where they're like, our first confirmed case was say February, I want to say. And they said, but we have all these specimens from before February. I don't remember when exactly they were able to identify spread, but they were able to move it back further than what they had previously thought based on current COVID tests.
Okay, but not all the way back before China, which would have been late December, right? So maybe January or something. I don't know. Yep.
So this is Larry out here in Seattle. The flu study picked up this stuff, the virus. I believe it was in January when they went back retrospectively and looked at the data that they had been collecting. But I believe those tests were actually prior to January. They had collected those specimens prior to January. The CDC shut them down at that point in time in terms of allowing them to do any further testing about that. And it's one of those decisions that you kind of wonder about pretty significantly.
Well, we'll get to talk about that at length, I think, actually, because it does get to exactly the topic of the talk, is how we balance these issues as we pursue research. So I'll go back to TJ. I just want to make sure I understood that I will do some little online research probably while you're talking, TJ. I don't know
that was helpful. So I wasn't exactly sure what data it was, but Larry's point was right, was the data was collected prior to it actually being a public health crisis. And so that was sort of the dispute is, are we allowed to change its status because there's now a pandemic? So all these 3 scenarios, though, or these 3 usage routes, have different implications for secondary usage. One thing I thought it'd be helpful to talk through a little bit, though, is why it matters whether it's public health or for research. This is something that is sort of ingrained in the thinking, and anyone who's thought about the regulations or read them. But it is kind of weird if you explain this to other people, why we have these different categories. So I think the regulatory and ethical framework, not just in the US, lots of places, but it's very explicit, I would say, in American regulations, is that we have these different domains. So data protection regimes are designed for different uses, and that has implications for consent and for oversight. So for example, we have clinical data, which is supposed to be for patients, QI, QA data, which is mainly, it maybe comes from patients, but it's supposed to be for the health system overall, you might say. Research data, which is for generalizable knowledge. And then public health data, which is supposed to be for tracking and controlling diseases. And so, of course, they overlap in certain areas, but they have their own set of rules and ways of thinking about why they're special. And the challenge, though, is that we have all these other rules or other sort of exceptions to turn this data into secondary research data, where, for instance, if you de-identify data, all of a sudden you can use it for secondary purposes, and all the special properties that they were supposed to have sort of go away. And this is where a lot of the ethical and regulatory challenges come up, because we may have been tracking that pipeline, say we have public health data, or we have data that was collected in a clinical setting. Now it's all just secondary data, and there are fewer restrictions. And so there's a lot of tension on moving from this initial context to the secondary context. But then once you get to that point, there are far fewer restrictions. And so that's where a lot of people are concerned, and where a lot of the questions come in. It's moving from that first case to the second. But so that domain way of thinking is very important, at least for a lot of ethical discussion. So the first question a lot of people will ask you is, what is the context for the initial use of the data? Was it for research or public health? And then what is your intention in your secondary use? And so usually the way this is defined is, what is the purpose for your use? If you watched Marriott's Treats talk a couple of years ago on QI/QA data, she talked a lot about how we identify what exactly is for research. And of course, it's very difficult because a lot of times it's the data we collect is for generalizable knowledge, but it's also for lots of other things. So there's no hard distinction. The common rule actually has some clarification on this. So the revisions that were made a couple of years ago made a clear distinction between research uses and public health surveillance activities. So public health surveillance basically has, or public health activities has two conditions. So one, it has to be for identifying, monitoring, assessing, or investigating public health signals, onsets of disease outbreaks, or conditions of public health importance, which a lot of researchers might say, yes, my data fits under that, but then you also have to meet the second condition, which is that the use has to be authorized by a public health authority. So if your researcher says, hey, yeah, my research has public health relevance, I'm doing that first thing, that's not enough. It also has to be authorized for a public health authority. So thinking back to those three scenarios I outlined, it's really helpful actually for, say, a research institution to partner with public health authorities, because you can say from the start, we are doing both research and public health. If we were in that first scenario where there is no research consent, you wouldn't somehow be able to avoid research oversight or research regulations by just saying, but my research, or the data I'm collecting is relevant to public health. You'd actually have to have a public health authority who's there saying, I've directed this to be for public health. If, on the other hand, you're intending to use COVID data for a secondary purpose, which is research, you will have to go through research regulations. So you have to submit to IRB. You can't just say, I'm doing public health activities, I don't need to submit anything for oversight. And so I guess the main takeaway, I should say, is that although there are no hard regulations around you can't move between public health and research, there is sort of a general consensus that more scrutiny is required if you're moving from public health to research in terms of domains. Not that you can't do it, but there's a general agreement that scrutiny is required. But so the rules, again, are not firm. The sort of guidance that the common rule requires or provides allows for exemptions. So if you've already decided in that first case, you are doing research, you're now submitting or planning to do research with COVID data, and you're submitting to the IRB, there are 3 exemptions that would allow you to use that data without getting consent. So Peter's last Streets talk talked about these in more detail. These are the 3 that are directly relevant to COVID, though. So again, just to make this clear, this is if you have access to COVID data, you now want to do research on it, And here are some conditions that would apply so that you can use that without consent, but it would require IRB approval. So one is if the information is already publicly available. So perhaps people have already posted their identifiable, not just COVID data, but other health conditions, and it's on some website for researchers to use. You could ask the IRB for an exemption to do research on that data. If the information is de-identified and you have no plans to re-identify it, again, you can apply for an exemption. And the last one is interesting. There's an exemption for data that was collected for public health activities and purposes under Hipaa. And this would include things like hospitals or health systems reporting their sort of aggregated, de-identified public health data to the authorities. So telling the county health officials, hey, we have 100 COVID cases today, and here are maybe their comorbidities or things like that. Or a lot of that information would already be de-identified, so the IRB would do a review and say it's fine for you to use this without trying to get those patients' consent. You only have to meet one of these conditions to be clear. There are some other conditions that are relevant, but these are the ones that are most relevant to COVID. So to sort of wrap up what I think are the main takeaways from things I've already said. So the first thing to think about if you're planning to use some COVID data for secondary purposes is figure out whether the initial usage and the second usage count as research or public health. And then relevant to what I just said is does the data contain identifying information? So if it's de-identified, you can apply for an exemption. This is just referring to data, not specimens. If your data does have identifying information, you'll either have to get consent from the initial participants, or you can apply for a consent waiver. People seem generally familiar with consent waivers, so I'm not going to go into details on that, but you can ask the IRB for a consent waiver given certain conditions. The third thing, which is really important, I sort of already hit on this, is that the consent form is going to really matter. So thinking back to the three scenarios, if you have contributed a COVID specimen for mitigation testing or whatever, it may have just been as part of public health. And it may say that in the consent form. We are just collecting this data as part of disease surveillance. You will not be included in any research study. If that's what people have signed up for, it would not be permissible then to use that data in secondary research activities. Consent forms don't always specify that. They may also, if they do specify what is permissible, what sort of research activities might be included, they might be very broad. So that's another thing to think about. The Seattle flu study, my understanding is the consent form said more than just flu. It said something like infectious diseases. And so that's where a lot of the attention came in, is what counts as an infectious disease in this context. COVID seems to fit. So not only do we have the authority for public health reasons to use this data again, but the consent actually said it could be used in other studies on infectious disease. I don't know, strictly speaking, if that was the case, but that's the sort of consideration or sort of thing you should take into account in thinking through secondary use. These are the main takeaways, I think, from the regulations anyway, and some of the official ethical guidance that's been given. To think about these three questions. Pertaining to that third one, though, there's one other thing that's relevant. So even if you do, say, have a consent form that does not prohibit research, so in any of the scenarios I laid out, if the consent form does not say anything about research not being allowed, and you then submit to an IRB, the IRB can still think about the risks of the sort of research you're proposing. So some of the things that people have discussed as being seen skeptically by our IRBs are things like, we have de-identified COVID data and we're going to compare it to other third-party data sets, especially because there are a lot of private companies that are also collecting COVID data. You can imagine how this would go. I have public health data from the county or whatever, and I'm going to compare it to, say, mobile data that we've collected from some third party. An IRB may look at that and say, yeah, sure, it's de-identified, but you're going to learn a lot of identifiable stuff about the citizens of our county. So that's my review of the main regulations. So the three main takeaways are these that I just outlined. I'm going to conclude, though, now in the last few minutes in talking about some of the ethical issues. They're all relevant to everything I've said so far, but they go beyond in a few ways. And the majority of them are going to be more skeptical sounding, or more along the lines of thinking why we should be concerned about secondary usage. But then I'll conclude on a more encouraging note. So one ethical concern a lot of people have been talking about is overwhelming consent disclosures. So COVID's been hard. For a lot of people, you may have had COVID, signing consent forms, in a hospital setting when you're very sick. You may also have gotten tested multiple times. It may have been different tests, a saliva test, blood test. It may have just been for mitigation testing, et cetera. There's a concern that people don't really understand what they're contributing to. And this should maybe lead us to be more concerned about using a lot of this data for secondary purposes. A related concern is around privacy. So especially last spring, when I anyway first started thinking about this, a lot of the rhetoric around research ethics during a pandemic was, hey, we should really encourage people to share data because this is important for stopping this as soon as possible. So especially around contact tracing apps, people are saying you really should be willing to do this because it's to the benefit of all of us. A lot of that has not been realized, but there have been lots of cases where people are sharing more data than they strictly need to for their own COVID diagnosis. And at the same time, there has been flexibility in regulations for good reason, but this could lead to a situation where people are sharing more data than they would otherwise want to have included in research. And there's a lack of disincentives for people to misuse that data. That's the concern. Related to that, and I already mentioned this, but mobile health data by private entities, as has been discussed in previous TREATS talks, is unregulated. So there's a lot of data that's been collected through mobile apps, not just for contact tracing, but for, say, tracking your own symptoms, or maybe you have opted into some sort of surveillance program, where those things are not really regulated. Researchers can sort of use them without having to go through any of the normal procedures to make sure that the data is being used responsibly. And my last sort of critical note is broadly going back to the idea of domains, or the domain-oriented way of thinking about ethics and regulation of data use, is that consent for public health is not consent for research. So in the revisions to the common rule, one thing that was changed in an attempt to prevent downstream uses of data was that you need to put in research consent forms now telling people whether their data could be de-identified and used for secondary purposes. That's now a requirement. You have to tell research participants either your data will be identifiable going forward, or we're going to de-identify it and maybe it will be used for other purposes. That requirement, though, is not going to be a part of public health consent forms. You can still de-identify COVID specimens or COVID data, and it will not include that sort of notification. So going through the public health consent pipeline, it's just less protected than going through the research consent pipeline. And that's likely not going to change in the near future. So just another reason to be concerned. My last encouraging note, though, sort of a countervailing consideration from these first four, is maximizing the value of the data and specimens we've already collected. So you might think the data is already out there. We've already collected a lot of data over the last year. Millions, hundreds of millions of specimens. There's no way of getting those back. So now we should really try to optimize the value of those. And optimizing the value means using it for public health purposes. We're of course not at the end of COVID. It may be with us for a while in some form. And so really we should be pushing for secondary use, you might think, but make sure that it's for publicly beneficial purposes. a lot of the debate about the use of other types of data, say clinical data for secondary purposes, will go something like this. You'll say, yeah, you can only use patient data for research insofar as the benefits of that research will then go back to the patients. It's a common argument anyway. You might make the same argument for secondary use of public health data. Sure, we can use all these COVID specimens that are running around, but only if it's going to actually help us, stop the current pandemic and maybe prevent the next one. So there may need to be more scrutiny around the sorts of uses that people are putting this toward. What that would look like, it's hard to see, but that's sort of one reason we might be thinking the opposite direction from some of these skeptical concerns I've outlined. With that, though, I am happy to talk about any aspect of either the regulatory or ethical concerns, or there's some other secondary usage that people have in mind for the COVID data we've been collecting. I think that would also be interesting. Thank you.
Okay, outstanding, TJ. Thanks so much. That talk excites me, as you know, from working closely with me, because it really gets at a topic we hear a lot from researchers, and which is obviously very much in the forefront of all kinds of biomedical research, is the use and reuse of data in a range of projects. Now the COVID case and public health data. Let me open it up to any questions or about this or related topics. I've got some specific in the COVID era, and then I've got some about de-identified issues. So again, questions from anybody, please. And TJ, you can call on your.
Right. So, hi, I'm John. I'm with Julie. I think you've already answered a little bit of my question. And Peter, you mentioned you might want to talk about de-identified data. Are we being a little disingenuous by saying it's de-identified data when you use a term like re-identify it? If the public is going to hear us say they've de-identified our data, that seems to give a message that there's a clear go-ahead to use it because it's never going to come back to me personally. But we live in an ever-increasing age where the power of our technology, if somebody wants to connect that back to a particular patient, it can be done if it isn't already doable. You mentioned you can re-identify the data. Perhaps the language needs to change. Perhaps it just seems to open up a Pandora's box there. And I was curious if anybody had any comments on it.
Yeah, this is something we've talked a lot about internally. Peter and I just published a paper on what are the risks of re-identification. And as you just said, it is pretty easy to re-identify data. Health data and data for research goes through various analyses to make sure it's not easily identifiable. But depending on the data set, it's hard to say what easy looks like, but it could, at least among data scientists and experts, they could re-identify a lot of the data sets pretty easily. And the common rule revisions did require a return to that issue, because that was a big debate, is should we now have a different definition of identifiable? They decided not to make those changes. And so I think, what was it? Within the first 5 years, every IRB has to return to that issue of, do we need a new definition of identifiable? I have not heard anyone proposing a new definition or a revision to the definition. I have wondered, though, with the almost inevitable challenges that COVID data are going to raise, whether that could spur people into, really motivate people into rethinking the definition. Because a lot of the debates have been around genetic data, and of course, the saliva and blood that people are providing has a lot of genetic data. Yeah, so I would also be interested if other people have solutions to that problem. That would be very helpful.
So Larry here, with regard to that last question, and again, I'm thinking of a large study like Seattle flu that's going on. All of the participants are are known to the investigators in that study. Is there a challenge in simply reaching out to people? And I'll use the COVID virus as the example right now. I think there's an awful lot of people that would probably say, yeah, anything you can do to help with this thing, it's okay to use my data. But I think you would have to reach out.
Yeah. And I suspect there's a concern that if you did reach out, not only would they say no to the secondary use, they could say, also, I'd like to withdraw from the initial study. I could see that being a concern. Relevant to that point, I thought about putting this as a case in the presentation. The All of Us program ran into that problem for exactly this reason. They had consented certain American Indian groups into All of Us and asked if they could use their specimens and data for COVID purposes. And that sort of flagged for them of, oh, you could identify us pretty easily. We didn't think you'd be able to do that. We thought our research data was aggregated, but they had basically been presented the possibility of, hey, we might be able to go to each one of you and consent you into a different thing. And that did lead to greater concern. I don't know if the participants in the Seattle flu study would feel the same way, but at least the All of Us participants, that's a program at the NIH that's collected a bunch of people's data and specimens. They ran into that very issue of trying to reconsent, hoping there would be that sort of motivation to contribute, but instead it led to concerns about, hey, how are you possibly going to re-identify us? We thought that was not going to be a possibility.
Yeah, along those lines, when something is, I guess, officially re-identified, it seems like you have this opportunity for kind of like playing checkers to kind of hop and skip in and out of identifiable, not identifiable, and somewhere down the road, yeah, I think a lot of people would say, yeah, you want to do something with public health with my de-identified data, great. But then if it gets identified officially, then it moves to a different set of rules and regulations, and then it gets unidentified. And I can see a scenario where down the road, that data might be being used by someone who originally had very good intentions to support public health. And now it's in a realm that they have absolutely no idea that it could have ever ended up there.
Yeah, there's a phrase, I don't think Jane is on, Jane Hartsock uses the phrase, it starts to look like specimen laundering. So in the context of biobanking, like we just keep going from one domain to the next. It's just a way of finding what new loopholes to be able to use people's data. It does get very confusing. So data scientists or data scientists or people who are experts in this area tell me that it's actually not that data is not that vulnerable, that there are ways of protecting this if you have good oversight, where it doesn't look so much like specimen laundering.
So
yeah,
something I want to talk about here is this is wonderful because we're getting at core issues in research ethics, and we always like it when these talks start very specific, very technical, regulatory, and we get to a big topic. And one thing's coming out in these comments so far, I would say to my ear, and again, that's just me because I'm a little hung up on this, as TJ knows, is that you sort of have this ideal that you can have everything. You can have your cake and eat it too, right? So you can have great access to data with full understanding of participants and continued conversation with them about how we're using it. And it's an ideal that is a beautiful world. And as TJ is saying, though, sometimes, I mean, always the real world lets us down comparatively. And you go back to people who talk about this, and they actually need to even have this conversation. There are all kinds of things going on that are not really about consent, but about education. So like data that's de-identified for research use doesn't mean that nobody knows whose data that came from, right? Regan Streif's, say the biobank, the India Biobank, the data is identified in the biobank. But if you give it to a researcher to do a study on some diabetes aspect, say, that researcher has no idea who it came from. They are given a de-identified data set, and the research they are doing is approved on the basis of being de-identified data. It's not sort of de-identified in the galaxy, but it's de-identified for that research and that research use. Now, that's a complex thing to explain to anybody. I'm not sure I did a good enough explaining it to us, and we are on this call a lot of people with expertise. And so imagine explaining that, and then take the step back and think of the ethical obligation. Say, well, look, it's hard to explain, but you better damn well explain it, and do the effort, make the effort to explain it and put to rest these worries, because what you're doing is you're ignoring people's, actual concerns that they will have if you talk to it about them. And if you just go forward and say, I can't tell them because they won't understand, they'll be anxious, then you're being the worst kind of paternalist. A part of me completely agrees with that. And a part of me completely rejects that and says, what you're proposing is a use of resources to educate the population about something which has absolutely no risk, or incredibly small risk compared to real risks in people's lives, to any other risk that any of these people are facing when their de-identified data, in the sense I described, is used for other research purposes, such as, COVID or other public health things, or maybe making progress in diabetes. So I'm just sort of throwing that out for consideration, that I think we have very understandable tensions here in the ethics world. On the one hand, we want people to know, as TJ may have, I don't think he mentioned in this talk, but maybe previously, and maybe I did too in previous talks, when you ask people what they want to be done with their de-identified data, they say, I am fine with using whatever the heck you want, but I want to be informed in that conversation. And that's, again, gets back to that ideal view. They say, I will say yes, but I want to be asked. But the problem is people don't actually say yes when they're asked. The expense of going to ask 1 million people for their approval for reuse of their de-identified data is actually a problem. So it is really a complicated area of ethics. It's a Necker cube. Where you can look at it one way and the other. And I'm fascinated, and I'm really share TJ's interest in sort of how are we trying to traverse these challenges. Feel free to yell at me, not TJ, yell at me. I just said that we shouldn't be asking people for consent. Larry, I see you've come off mute. You're gonna yell at me.
No, not at all. I was thinking about the original consent forms, or the original conversation about consent when bringing people into a study, and acknowledging that, there might be a future need to come back to you and say, boy, we've got this wonderful data, and we'd like to look at this too. And I'm trying to think back on the consent. I'm in the Seattle flu study. They made it available to all of us at the university. I'm trying to think back. I know there was something in there that basically said, we might want to use this data in the future. Acknowledged that, and I think they also said, we would let you know if we were going to do that to reconsent. But I think this is in part a huge trust issue, and I think you can maybe diminish that trust concern by at least having some of these conversations up front. Now, if your consent form is 30 pages long, it's kind of hard to do that. And I recognize that. I think this is more form of conversation than it is just the written form. So my thoughts.
Other thoughts?
Could you turn off screen sharing so we can see everybody?
Yeah.
Yeah, although see Larry, I'm gonna totally undermine that, because I wanted to go back. Well, actually, wait, maybe you could speak first, TJ, and then if you need to do screen sharing, feel free. But you mentioned that there's a new requirement of disclosing the possible de-identification of data, and then it's used for secondary research. Is that now? I'm not sure I'm very familiar with that specific rule of the new common rule.
Yeah. Hold on a sec. I'll put it in the comments. Yeah. I think I'll be telling your class, what is it, in 2 weeks about this. Because that was supposed to address some of the classic cases of like the Hipaa case, and other cases where people consented to research, and maybe they had been told, as Larry was just suggesting, that secondary use was possible, but they may not, there's a lack of details. And so the requirement, as I understand it anyway, is you have to tell people it's going to remain identifiable in the form you've just given us for our research, or we could de-identify it and share it with lots of other people. And that goes along with the requirement of you have to tell people that it could be shared with commercial entities to at least give some awareness. But as Larry suggested, so that might address some of the trust concerns. But at least when talking about this with students, a lot of students will say, that's not a whole lot. Like, so you've told me it could be used in something, but then it's out of your control. That makes me concerned.
And the explanation of what de-identification is not part of it. It's just like we maybe de-identified, you know what I mean, wink, wink, which does not mean that nobody knows who you are. It just means the people who will then use your data will not have any easy way of re-identifying you, although it is possible. And by the way, it's possible, which would be a full disclosure, which would probably raise more questions than it would solve. And this is always the case when you say something in this world that you think you're reducing uncertainty, but then by speaking, you actually increase uncertainty because you say things that sound like, wait, what did you just say? That doesn't seem to make sense from what I understood. Larry, you're back on camera. Or you're just on camera to visit, not to speak.
No, I'm just agreeing. I'm agreeing with you. I put myself back on so you can see me nodding my head here.
Yeah,
You know,
I like that.
the complex issue. It's a complex issue, and TJ's already acknowledged the untoward effect of trying to do that with the American Indian population. Again, I don't. It's an ideal almost to say, well, if you just said something to them, had the conversation up front about what you might be able to do in the future. But holy cow, how much time do you have to do consent? And like I said, if it's buried in 30 pages of consent, it's probably not gonna be visible.
We often talk about this as a burden on the researchers. Like, oh, we don't have money, we're spending our money on research, not on talking to people at great length and educating them. But actually it's a burden on the participants too. Like, I don't actually want to learn about de-identification processes and these mathematical models that people use for triangulating my identity. I could care less. I'm just trying to give you a gift. In fact, some of the research we've been doing at the Center for Bioethics, led by TJ, has to do with asking people, what do they want to know? What do they want to be educated about? Because on the one hand, we want to educate them to treat them fairly. On the other hand, what we're doing is adding a burden to their very simple say, like, oh yeah, sure, to keep me safe, but let me contribute to this research. And so again, I can talk for hours, and I won't for a change. I will not go on forever. TJ, anything else we should know at this point? Either this is stimulating you or any take home points beyond what you just said? I think you gave us good messages already, but anything?
The other thing, the trust thing, we've talked about a lot. I've wondered, so there's a concern about lack of trust in research if people find out you've used the data for other things. I don't know if I've seen a lot of research on potential effects on trust from secondary use of COVID specimens. So what would people think if you say, yeah, we shared your data with researchers at IU who have now done a whole bunch of other cool studies on your COVID data? I imagine that people would be into that, but I don't know.
Okay, well, thanks everybody for coming. Any other questions? This is your chance. I hope you found this helpful. Again, that was thumbing up. I mean, the video and the PowerPoint will be posted on our treats page. I sent that in the link. TJ's coming back on now. Go ahead, TJ.
I realized I put in the that common rule requirement in a direct message rather than a chat to everyone. So here now is there you go. For people who want to punish themselves by looking up the common rule.
Fascinating. So if it's identifiable, the initial limits include a statement, either indicating that it will or will not be used in secondary research. Does that mean whether the identifiable data will be used in secondary research?
No, either it'll remain identifiable in the study, or which means we're not going to share it with other people, or we could de-identify it and use it.
So I would think I was actually, one thing, again, I don't want to extend us too long, but what you said before was if there's a consent that says specifically that the data will never be used for additional studies, then the IRB is not going to allow you. And obviously ethically, you could say, Peter Schwartz, all your concerns about the vagaries and complexity of consent notwithstanding, it's not ethical to do something you explicitly said you were not going to do. It sounds like as a researcher, you should not put that line in.
Yeah.
Unless you really think the research participants want or need it in order to participate, reasonably in your study. Don't throw that line in unless you've got a really good reason to, because even if you say, well, they didn't rely on this promise, they didn't understand this promise, you make a promise.
Yeah.
It's not exactly ethics, graduate level to say you probably should follow it.
The commentaries on the Seattle flu study have really hit up hard on that point of be careful what you're promising to participants because it could hurt you later on.
They said they would not use it for anything else.
So my understanding is that they said they would use it for other infectious disease research.
And so there, oh, by the way, and then a perfect closing comment. I did look up, if maybe other people on the call did. The circulating COVID in Seattle was in December.
December.
It apparently doesn't, from the Seattle Times reported, I can't give you anything, better than that. Which of course raises my question of, do we know the origin of the COVID virus at all? If we start saying it was here in the US in December, was it just the first outbreak that happened in Wuhan? And that of course raises all kinds of questions, but I cannot give credence to any of that. I just was reading an online Seattle Times, which seems to be reliable. Larry, do you want to help me understand where COVID came from? Because I was told that it came from Mars. That's what I heard, so.
Yeah. I will not comment on the Seattle Times being reliable. It is and it isn't sometimes, just like anything.
Exactly.
What was the date on that article? Do you remember, Peter?
Don't. It was a quick surf. Let me take a look. What does that do? My
Because all I was going to say is no question that it was December data, but there's actually some suggestion that it may go back into November as well. Yeah. And of course, our patient one here that was identified was a patient that came from a gentleman that came in from Wuhan directly and was affected. But that was just the one, that was patient one that was identified later, with the disease, not somebody that was potentially affected earlier and thought it was flu.
In January, that was a January case, right? So This is a story from December 1st, 2020. And again, it's reporting that a year earlier in December 2019, there were 38 or something, a significant, 84 samples were found to have neutralizing and activity specific to SARS-CoV-2. So that is fascinating and very mind-boggling. Okay. Bye, guys.
Thank you all.
Thanks for being here. Take care. Thanks, Larry. Thanks,
Bye-bye.
DJ.